Hello everyone, today we will discuss Plasmodium Vivax life cycle in mosquito. Plasmodium Vivax life cycle in mosquito. So this life cycle contain 4 stages. First one gametogamy, second one fertilization, third one formation of oocyte and oocyst, fourth one sporogamy. Previous class we will discuss sporogamy. Gametogamy means the formation of male and female gametes from the gametocyte. Formation of male and female gametes from the gametocyte. Male gamete formation and female gamete formation. First one, male gamete formation. The nucleus is divided into 8 daughter nuclei. The cytoplasm, the formation of flagella-like structures. 8 flagella-like structure. One nucleus enter into one flagella. So they are the separating from the cytoplasm. So this process is called as exflagellation. Totally the formation of 8 male gametes. Next one, female gamete formation. So the female gametocyte undergoes few changes. They transform into the female gamete. So this process is called as maturation. Next one, the nucleus spots in the peripheral inside is like a projection is called as fertilization cone. So they are the male gamete formation and female gamete formation. Second side heading fertilization. Second side heading fertilization. Fertilization. Second side heading fertilization. Fertilization means the fusion of male and female gametes. Fusion of male and female gametes is called as fertilization. Fusion of male and female gametes. Called as fertilization. The fusion of male and female gametes. Fusion of male and female gametes is called as fertilization. Fusion of male and female gametes is called as fertilization. First step. So the fertilization process. So they are the male gamete. So they are the female gamete. So they are female gamete region. The flagella, like this gamete, male gamete. So they are the male gamete. These are entering into the female gamete of fertilization cone region. They are entering into the fertilization cone region. So these are formation of zygote. These are formation of zygote. So called as zygote. Zygote. The fusion of male and female gametes is called as fertilization. So the male gamete entering into the fertilization cone. The male gamete is the locomotion. So the actively moving a male gamete, they reaching to the female gamete site. The female gamete actively moving male gamete reaching the fertilization cone of the female gamete. So they are entering into the female gamete. So this is called as formation of zygote. This is called as zygote. The formation of zygote. So they are dissimilar gametes. So they are called as anisogamy. This fertilization, dissimilar gamete. Male gamete is separate and female. Structurally different structure. So they are the anisogamy. This fertilization is called as anisogamy. Next one, the zygote is round and non-motile. The zygote shape is round and non-motile. Round and non-motile. Round and non-motile. The zygote, they are the round and non-motile. They are the zygote. So they are the round shape and non-motile. So they are the fertilization. Simple fertilization means fusion of male and female gametes is called as fertilization. So the actively moving male gamete reaching to the female gamete fertilization cone region. So they are entering into the female gamete. So they are formation of zygote. So they are the dissimilar gamete fusion. So they are called as anisogamy. Next step. So these are the crop wall basement membrane. So they are the crop wall basement membrane. These zygote So these are the basement membrane of the crop wall. So they are the basement membrane of the crop wall. Crop wall basement membrane. So these are called as zygote. So these are the zygote. Fertilization time, the fusion of male and female gametes, the formation of zygote. The zygote is round and non-motile. Next one, the next step, formation of oocyte and oocyst. Third heading, formation of oocyte and oocyst. Formation of oocyte and oocyst. Formation of oocyte and oocyst. Third heading, formation of oocyte and oocyst. So the zygote, these are sometimes after few hours they are transformed to the slender shape. They are transferred to the slender shape. They are transformed into the slender shape. Slender shape. They transform into the slender shape. The slender shape and motile. Zygote, they are transferred to the slender and motile. Slender and motile. So they are the actively moving. So these stage is called as oocyte. So this stage is called as oocyte. This stage is called as oocyte. So the zygote, they are remain sometimes active, so they are actively moving and their shape is a slender shape. So they are called as oocyte stage. This stage is called as oocyte stage. So these oocyte stage, they are persist the crop wall. Persist the crop wall. So they are coming the outside the crop wall. So they are coming the outside the crop wall. They are formation of a round shape. They are transformed to the round shape. These are changed into the round shape and surround the cyst wall. Surround the cyst wall. Surround the cyst wall. So this stage is called as oocyst. This stage is called as oocyst. This stage is called as oocyst. Simple. So the zygote, round shape and non-motile. So after the 18 to 24 hours, these are converting to the slender and motile shape. So they are the persist the basement membrane of the crop wall. Means so they are rupturing of the crop wall of the mosquito. So they are coming to the outside. Come to the outside of the crop wall. So they are the crop of the mosquito. So they are coming from the outside the crop wall. They are transformed to the round shape, transform to the round shape and they formation of around the cyst wall, So this stage is called as oocyst. This stage is called as oocyst. So the formation of oocyte and oocyst. Formation of oocyte and oocyst. Fourth side heading, sporogamy. Fourth side heading, sporogamy. Sporogamy. Sporogamy means the formation of sporozoites. The formation of sporozoites is called as sporogamy. The formation of sporozoites is called as sporogamy. So next step, the oocyst stage, the nucleus, the oocyst stage, the nucleus, reduction division. So the nucleus is according to the Bano. So there are the reduction divisions. Many first reduction division after many mitotic divisions. Many mitotic divisions. After is called as pronuclei. Next step, each nucleus surround the each nucleus surround the cytoplasm. Each nucleus surround the cytoplasm. Each nucleus surround the cytoplasm. Next one. So these each nucleus and surround the cytoplasm, they form a sporozytes. So these are called as sporoblast. Next one, their formation of sporoblast. Inside the oocyst, they contain a sporozytes. Inside the oocyst, they contain a sporozytes. So this stage is called as sporoblast. This stage is called as sporoblast. Sporoblast means, so they are contained in the oocyst. So oocyst, inside the oocyst they contain a sporozytes. So they are called as sporoblast. The final step, the sporoblast is rupturing. The sporoblast is bursting, releasing the sporozytes into the salivary glands. So they are the salivary glands. Mosquito salivary glands. So these are sporozytes. So they are the sporozytes. So these sporozytes are releasing to the salivary glands of the mosquito. Next one, these are migrate from the salivary glands to hemocoel. So they reaching to the first step, reaching to the blood circulation. The blood circulation to reaching to the salivary glands of the mosquito. So these mosquito bite a new human beings. This saliva to sporozytes entering into the another human being. So they are called as female Anopheles mosquito in the Plasmodium life cycle in female Anopheles mosquito. Simple. So they are the final step. They are releasing to the cryptozytes. So they are entering into the blood circulation to reaching to the salivary glands of the female Anopheles mosquito. So they are the female Anopheles mosquito. The Plasmodium life cycle in female Anopheles mosquito. So these are the scientist RAS. So they are the crop of the mosquito. They contain a 50 to 500. So they are the nodules like structures. So they are the oocysts are presenting on the crop of the mosquito. 50 to 500 in number. Identify the RAS. Identify the scientist RAS. So they 50 to 500 small nodules like structures. Oocysts the presenting on the crop of the mosquito. So they are identified the scientist RAS. So they are the 4 steps. Fertilization, the first one sporogamy, first one gametogamy, second one fertilization, third one formation of oocyte and oocyst, fourth one sporogamy, final step releasing to the sporozytes. So they are called as sporozytes. So they are the life cycle of Plasmodium in female Anopheles mosquito. Next one, prophylaxis. So this prophylaxis using to the DDT, dichloro-difluoro-trimethane, and BHC, benzene hexachloride. So they are the spreading to the mosquito areas. So they are the killing to the mosquito larva stages. Next one, they are the introducing to the larvaceous species. Like Gambusia fishes. So these Gambusia fishes they eating to the larvae of the mosquito, Next one, insectivorous plants, introducing to the insectivorous plants. So they are the eating to the Plasmodium, Vivax and the mosquito larva stages. Next one, they are the prevention of the mosquito bites using to the mosquito nets and all out good night, like, so they are the prevention of the mosquito bitings. Next one, they are spreading to the kerosene and oils from the stagnant water storing places, so they are spraying to the kerosene and oils, so they are killing to the larva stages of the mosquitoes. So they are the female Anopheles mosquito and the Plasmodium vivax life cycle in female Anopheles mosquito.